We're very fortunate to have Dr. Phillips as a Porphyria specialist and as a member of the UPA Scientific Advisory Board. Dr. John Phillips has been working in the heme biosynthesis field at the University of Utah School of Medicine for the past 30 years. The primary focus of the lab is biochemical analysis of the proteins in heme biosynthesis, including regulation of expression, metabolic interactions, and structure function studies. Dr. Phillips, thank you so much for being on here today and for imparting your knowledge to us. The time is all yours.
Great. Thank you very much. That was a great introduction. Before I get started with all the technical stuff, what I wanted to do was to thank the UPA really for, providing this platform for myself to get my message out, but also providing a platform for, the community to be able to come in and find out what goes on sort of behind the scenes, I guess, if you will, from the biochemistry side of the disease, and see really, what's going on more at a molecular level. So I think probably the easiest thing for me to do, because I am a professor and I have been a professor for way too long, the easiest thing for me to do is to start off with a couple of slides so that we're all on the same page, okay? Absolutely, anytime you need to ask a question, just put it in the chat. Somebody will bring it up to me. I'll stop. I'll try to figure it out. Don't hesitate if there's something that is unclear. Just let me know. I'm happy to go at the problem a different way. So I think the best way to get started, as I said, is to I'll share this screen, and you can see here. This is the biochemistry of heme biosynthesis. And I use this slide when teaching students because what it does is it puts everything sort of in a perspective. I think a lot of people say, oh, I have porphyria, but really what you have is you have a very specific type of porphyria. So what's shown in this slide, here in this first column, where underneath substrate, it tells you the biochemistry of how this happens. So the first step, for example, is the condensation or the molecular hooking of glycine to succinyl-CoA to make aminolevulinic acid. So it's ALAS is the enzyme, 5-aminolevulinic acid synthase, and the disease associated with having a defect in that is X-linked protoporphyria. Okay, and I'm not going to go down through every one of these because I have way too much to say about these, and I really want this to be more of a discussion among us. So look at this slide. Many of you have aporphyria, for example, acute intermittent porphyria. We've already heard about that from Lena, so there's a defect in this step right here, okay? A couple of things I want to point out on this slide. So the size of this arrow, you see the size of this arrow here in this column, tells you how much of that enzyme is present. And so you see this first step right here, ALAS. The arrow's pretty small. And the reason it's small is because this is the rate limiting step in the pathway, okay? And when I say that, rate limiting means the amount of this enzyme defines how much flow. It's sort of like how big is the garden hose, right? Is it a tiny little hose or is it a big giant fire hose? And this enzyme is what figures out how much can flow through the pathway. And there are 8 steps. And there are eight, we say there are 8 committed steps. And there are 8 committed steps because um, these enzymes don't do anything else. They only make heme. That's all they're interested in doing, right? And you see heme is down here at the end. And you see this little red dotted line that comes back here. So there's a feedback loop. And when you have lots of heme, it tells this enzyme to slow down. And when you don't have any heme, it tells this enzyme to speed up. Okay? And um, that becomes important when you look at managing the symptoms of the disease. So again, the substrates are here. Uh, the size of the arrow tells you how much enzyme there is. The name of the enzyme is here, and the disease is over here. The other thing I want to point out from this slide, there's a lot going on here. It's a good general slide, but I kind of have this arrow here, and it goes from yellow to brown, and it goes from yellow to brown for a very specific reason because these early pathway intermediates, they come out in the urine. But as the molecule becomes more complex down here at the end, those things come out in the stool. And it's important to understand this because a lot of times the questions I get from people is, well, how come I have to have a blood test for this? Well, you have a blood test for it because um, anything that's in your blood eventually gets filtered out into your urine. All right. Stuff from your digestive tract or from your liver comes out in the stool. That's sort of the physiology of how that works. Okay. So uh, on the next slide here, this is the actual biochemistry. And I don't want people to to try to memorize any of this, but these are the actual molecules. This is glycine. Glycine is an amino acid, and succinyl-CoA is a metabolic intermediate found in the mitochondria. This first condensation by ALAS makes this molecule here ALA. Um, and two of these come together. I kind of tried to set them sideways a little bit so that you could see that how these two molecules come together to form PBG. 4 of these come together to form this, what we call a linear tetrapyrrole. And then this bond forms over here to make uroporphyrin 3. And then you begin to change these acids. There's an acetate and a propionate here, and you don't need to know what that means other than it's biochemistry. But these get changed on the outside. And as these change on the outside, as the enzyme works on that to do various chemical reactions, it makes it less water soluble. And then these are the oh, sorry. These are the molecules that end up in your that come out in they go through your liver and they come out in the stool. Okay. But what's important really to recognize here is the these molecules here in the blue circle. Those are the ones that we really measure in the blood for diagnosis of certain porphyrias. These molecules here, These molecules here in sort of the purple color, the reason they're in purple is because those are the molecules that become fluorescent. And it's the fluorescent properties of these molecules that lead to the photosensitivity. So I think probably the best example is here in protoporphyrin. You can see there's two lines here and one line here and two and one and two and one and two and one and two all the way around the circle. I won't go all the way around, but there that alternating single double bond that goes all the way around the circle, um, that's what gives it the fluorescent property. Okay. And so when these molecules become fluorescent, they are the things that cause the light sensitivity. So that's kind of a general backdrop of what heme biosynthesis is. And now I'm going to stop sharing and I'm gonna see what kind of questions you guys have.
Yeah, there was a question on the first slide. What do the different colors of the boxes under the enzyme columns represent? Is it acute versus cutaneous?
Yes, actually it was. Yep. And um, so um, when we talk about acute, what we mean is we get this sudden onset of symptoms. And when we say cutaneous, what we mean is those molecules that build up that give you that photosensitivity. Those molecules they're phototoxic, they're in your blood, they're gonna get filtered and either get filtered by the kidney and come out in the urine, or they're gonna get filtered by the liver and they're gonna come out in the stool.
Okay, great. I have a question. So in AIP, the urine turns purple and starts to fluoresce, right? And why why does that happen, first of all?
Well, let me go back to share here. I'll go back here and share this. So that's a good question. So you see the third molecule over here,
Mhm.
PBG, porphobilinogen. Um, the enzyme that you don't have enough of is PBGD, porphobilinogen deaminase. You don't have enough of this enzyme. So PBG builds up. And it builds up to the point that actually it can make, it can spontaneously make hydroxymethylbilane, this linear tetrapyrrole. It can make that all by itself. You don't need an enzyme. It's a slow process. Um, uh, it 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 isn't immediate. It's a slow process. Um, and then what happens actually is this bond gets formed here, and we're only looking at the three isomer, but you see over here where it says there's this hydroxymethylbilane, non-enzymatic uroporphyrinogen 1, coproporphyrinogen 1. So what happens is non-enzymatically, these molecules are formed, and that's why um, that's why you see a change in color. But it
So
doesn't
my
happen, it doesn't happen right away. It
My
takes
follow-up
some time.
question to that is if that reaction can happen spontaneously in the urine, why does that not happen in the blood, and why are AIP patients not light sensitive?
That's because your efficiency of removal
Oh,
of
got it.
those intermediates is really pretty good. I mean, you're is if your kidneys are doing their job, which they're very good at that, They
Mhm.
remove those compounds and put them in the urine, and you don't have them. That's why.
That sense. Thank you. The next question is, please, is it possible to have biosensors which can be used at a point of care as a molecular assay in real time? Uh, what?
All right. So
So, go ahead.
let me just sort of separate what's going on here. So real-time PCR is what we all remember probably from COVID. Oh, I'm going to go and get my PCR test to figure out whether or not I have COVID. Um, that is a, a molecular test, but uh, it's looking for nucleic acid. It's looking for DNA, or it's looking for DNA that's made off of RNA. It's and PCR, you take DNA primers and you amplify logarithmically a piece of DNA, a small piece of DNA that you can then put a tag on, and that's why that is. Now, for a biosensor, there is the ability for some of these molecules, PBG, for example, to be detected in urine in the emergency room. The professors and doctors of the Porphyria Consortium, this test used to be available probably, I'll say 10 or 15 years ago. That test was available at some emergency rooms. But with the advent of, you know, changes in healthcare and everything else, that test has sort of gone away. And the reason it went away is because it's so very rarely used. In order for ER physicians and staff to be deemed competent at performing that test, it got too cumbersome for emergency rooms to have that test. The people that work on Porphyria, your doctors, the doctors in the PC, and those that support the UPA, we all want that back, but because of legislation and lots of other things, it's extremely difficult to get that test. But there is a like it's not really a biosensor per se. It's a test. It's a modification of a thing called the Watson-Schwarz test, and it makes a color change when it sees that PBG in the urine, and it's super effective. But again, because it's very difficult um, to meet the clinical standards of proficiency. It, it that's a hard question to answer, you guys. It's all rules and regulations that we don't like, but we have to live with.
Thank you. Which substance in the pathway is the most light reactive in terms of energy?
That's a good question. Um, so let me go back to share here. Hold on. Let me go back to share. And we'll go to this next slide. Um, so um, the so fluorescence is defined as uh, the change in energy of a photon coming in at one wavelength and leaving at a higher wavelength. So, for example, the 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 light that causes the phototoxic reaction is light in the 400 nanometer range. And each of these compounds here at the bottom, sort of all of these in the purple here, they they all absorb light at about 415 nanometers, but there's a couple of wave numbers different in each of these, okay? And then they all emit light around 620, 618, 620, plus or minus a couple of wave numbers. Um, so the one that has the most energy is the one that has uh, the biggest difference between the light that comes in and the light that goes out. Um, so that's called the Stokes shift. And I'll be honest with you, I don't have that number on the top of my head, but if I were to guess, I would say the biggest Stokes shift comes from protoporphyrin. Um, that, and I don't know why I remember that. I don't know exactly what wavelength for all of these. There's a book that I look at when I want those numbers. I go to this there's a big fat book of porphyrin chemistry, written by a guy, uh, Dr. Smith down in LSU. There's a big fat book, and I just go and I look at those numbers when I need them. I don't have them memorized. I'm sorry, but I'm pretty sure it's protoporphyrin has the biggest Stokes shift.
Great. Thank you so much. The next question is, on the first slide on the heme pathway, at the bottom, you're showing heme and mentioned that if you can't process it, it sends a signal back to the start of the process to make more. With porphyria, if you can't process heme correctly, would this mean that you are constantly in the cycle of making heme? And how does that process affect toxin buildup in the body?
Okay, so let me go back again. Let me go back here. Uh, because that is a really good question. All right. So yes, the issue is coming down through this pathway. When you have plenty of heme, it signals back to make less. Um, primarily, I'm going to tell you primarily in the red blood cell. Okay. 85% of the heme that you synthesize every day, 85% of it goes for making hemoglobin for your red cells. Another 10% or so goes to making heme for enzymes in the liver. Heme is a very important cofactor for cytochrome P450s, and cytochrome P450s are those enzymes in your liver that break down toxins. For example, I'm having a I'm having my coffee right here. P450-1A2 um, metabolizes caffeine. So there are P450s. There's oh I'll say there's in excess of 100 different P450 variants that we have, right? And those variants all process some some chemical intermediate to make it less toxic or make it more soluble to go out in the urine or make it more soluble to go out in the stool. That's what these enzymes do. And that's why the liver needs that heme. Okay, so now we understand where heme is used primarily. And the other 5% is in like myoglobin for muscles. Every cell, actually every single cell, um, makes a little bit of heme or or pulls it out of the circulation. We need heme for multiple different biochemical processes within cells. Um, I just gave you a couple of the big examples, right? Hemoglobin and cytochrome P450s. Um, so when you're trying to make heme and you don't have enough of it, it does signal back to this to make more of this, which in turn floods more down through the pathway. So in effect, um, those people that are looking for drugs to treat porphyria, they're looking for drugs, for example, to slow down this first step in the pathway because this is the rate limiting step. When you don't have enough and it's not signaling to shut this down, this keeps going full speed, just keeps going full speed, more and more and more. And if they can slow that down, then they can get less of these intermediates in the pathway. Hopefully that answered your question. If it didn't, ask it in a different way and I'll try to answer it in a different way.
Thanks so much. Has there been any research on fixing our mitochondria since it plays a major role? This reminds me of the Dr. Terry Walls and the Walls protocol.
Um, yeah, so um, let me again, I'll go back to sharing. Sorry, I have to keep jumping back and forth here. Um, but one of the things that's really important to point out. So if I were to uh, if I were to if I were to draw a line right here, doesn't let me do that. If I were to draw a line here, so succinyl-CoA and glycine, that's in the mitochondria. It forms ALA. ALA is transported out of the mitochondria. All of these steps here to PBG, to HMB, to urogen 3, um, those steps happen in the cytosol of the cell. Coproporphyrinogen 3 goes back into the mitochondria, and these last steps happen in the mitochondria. Okay? So when you say fix the mitochondria, there's um, the the genes that encode the enzymes are all they're all nuclear genes. Your mitochondria actually has its own DNA, but the DNA that's in your in your in your nucleus, that's the DNA that has the the coding regions for all of the enzymes in heme biosynthesis. So the enzymes get made in the cytosol, um, like ALAS gets made in the cytosol right here. This gets made in the cytosol and it gets transported into the mitochondria, that protein. There are regulatory steps that affect how rapidly this protein or how easily this protein is imported into the mitochondria. Coproporphyrinogen oxidase, CPO down here. That's also associated with the mitochondria. Uh, PPO, protoporphyrin oxidase, that's in the mitochondria, and FEC. It turns out that PPO and FEC are they actually are there's a gal that works in Georgia um, that showed that PPO and FEC, she says fetch, but I say FEC. PPO and FEC form a complex in the mitochondria. So again, to get back to the question, fix the mitochondria. So unless there's I don't know of any specific defects of mitochondria that affect heme biosynthesis. So if your question is, I'm not sure what you want to fix in the mitochondria. The mitochondria itself isn't necessarily broken.
Mm, Thank you. That makes sense. Next question is, why are some porphyrias tested in the urine and some in the blood or feces?
Because that's where those things accumulate, right? So yeah, let's go back here and these intermediates up here, ALA, PBG, hydroxymethylbilane has an extremely short half-life. We can't see that. We it's it 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 closes, it self-cyclizes non-enzymatically to form the one isomer so much faster than we can see it. It we for the sake of argument, unless you're doing this reaction in a laboratory where you can actually stop the reaction and isolate that intermediate in people, we don't see it. Okay. Um, Once you get to uroporphrinogen 3, that's that's present in the urine. So everything from here up, all of these porphyrias, you're looking at the urine. You can see these in the blood as well, because they accumulate in the blood and they're filtered into the urine. Um, but you can see these in the blood and in the urine because they are water soluble. But from here down, these coproporphyrinogen to heme, those are not water soluble. And so if it's not water soluble, then it ends up being processed through the liver, and it comes out through the bile duct and gets pushed into the stool. Why that is because if we look at the molecular nature of these compounds here, so all of these, you see all of these, you see this carboxylic acid, this oxygen and this hydroxyl group here. These make these very water soluble. And so an acetate and a propionate, an acetate is CH2C double bond O bond OH. That's an acetate. And a propionate, I don't want to get too technical here, but is CH2CH2C double bond O bond OH. So the difference is just one carbon in the length of that chain. That's why this has 2 lines and this has 3 lines. 1, 2, 3 carbons. So 1, 2 carbons, 1, 2, 3 carbons. That's the way that is. But they have this carboxyl group on the outside. So there's 1, 2, 3, 4, 5. Oops, sorry. 1, 2, 3, 4, 5, 6, 7, 8. There's 8 carboxyl groups on the outside of that. That makes it very water soluble. So uroporphrinogen decarboxylase, the enzyme that's defective in PCT, that enzyme cuts off the 4 acetates and makes them methyls, which is a CH3. It cuts off, you see, it cuts off 4 CO2s right here. It's cutting off 4 of those carboxyl groups, what we talked about up here. It cuts off 4 of those. 1, 2, 3, 4. It cut 4 of those off. And that begins to decrease without those 4 carboxyl groups. It's less soluble in water and actually comes out in the stool. And then again, this reaction, you'll see this arrow coming off this way, kind of pointing up. That means that's what it liberates. So CPO liberates 2 CO2s And so it makes a vinyl here and a vinyl here. So it gets rid of those two. And then you're left with only two carboxyl groups on these propionates here. And this is very, very hydrophobic. Um, these compounds, protoporphyrin, really, it's a very sticky molecule. It sticks to membranes. So when we do work on these things in the lab, we have to be very careful because a lot of when we're trying to characterize these things, a lot of what we do, it sticks to glass, it sticks to plastic, it sticks to everything that is not water. So I hope that answers your question.
Thank you. The next question is from someone who says, I have EPP. Which is most accurate for finding out how excessive protoporphyrin 9 concentration is for a person with EPP above average human levels? So, is a blood test or a feces test better?
Um, so the I will argue that the blood test is probably the important test for you. And the reason the blood test is important for you is because um, I think we need to bring this in context of where is the problem? Now, um, you have a defect in ferrochelatase. That's the defect in the enzyme, right? Um, but remember, we said that most of the heme that's made is made in the red blood cells. And so uh, those red blood cells, the first thing they do when they get excess protoporphyrin, there's a transporter that pushes it out of the red blood. The red blood cell doesn't want it. It pushes it out into the plasma, the fluid that is part of your blood. The non-cellular component of your blood is called plasma. So it pushes that protoporphyrin from the red cell into the plasma. And that gives you, the reason I say test the blood is because that gives you a very direct measure of um, what what your red cells are are trying to deal with. They they can't move it through the pathway, so they push it out. You can measure it in the stool, absolutely you can. But remember, that's after it's gone into the liver's processed it, and then pushed it out in the stool. So um, while you can see that, I think the most accurate thing to look at is the levels that are in your plasma. That's what your doctor's concerned with. That's what you should be concerned with because the level in your blood leads to how photosensitive you are. The more you have in your blood, the more photosensitive you're going to be.
Wait, that was actually another question. Are there any studies for protoporphyrin levels in EPP showing a higher number, meaning or correlating with a more sensitivity to light?
Yes and no. It gets a little bit tricky because um, we all have a little bit different color skin,
Mhm.
right? Um, and so um, somebody that might not have a very high level, but is of uh, English ancestry, I'll say, that has, very white, pale skin, like me, that might be, a problem. As many of the people with EPP know, one of the treatments for EPP is actually um, compounds that induce melanin synthesis Alpha MSH is one that is marketed. And what does that do? That kind of gives you a tan, and that tan helps protect that melanin that's in your skin, the melanin that turns in your skin, turns your skin brown when you get a tan, absorbs some of that 400 nanometer light that we talked about, and prevents you from being super photosensitive. So getting a tan, according to many patients with EPP, is helpful for them. I think the the people that are more fair, it's difficult for them to get over that initial boundary to get that tan that's going to help them, and that's why the stimulation of melanocytes with alpha MSH, so not through sun, but through a chemical reaction is beneficial. Does that? I hope that answers your question.
Thank you. The next question is, do porphyria patients have enough heme? And if so, how?
Uh, yes, they have enough heme. Generally, I will tell you they have enough heme. There are um, some people that don't make enough heme. People with CEP, for example. Um, if we let me go back here. Let me go back here and so you see the size of the arrows here?
Mhm.
That's how much enzyme you have, right? People that manifest CEP, um, they have been shown to have about 10% of enzymatic activity, okay, before they actually become uh, sensitive to the point that they go uh, and see a doctor. If you have if you have half the amount of uroporphyrinogen 3 synthase, you're fine. You don't you don't have the disease. UroD, for example, many people have a defective copy of in one gene for UroD, and they never have a problem in their life ever. It's only when something happens and to their liver do they ever really manifest the disease. Um, so um, I think when you're thinking about not enough, um, you got to remember that um, because heme is you, you need heme for red blood cells for hemoglobin. You need heme for detoxification of things in your liver. I my wife and I had dinner last night. I cooked. Um, I opened up the fridge and I saw that there was broccoli in there, so I made a stir fry with broccoli. Broccoli is, as many people know, is called a cruciform vegetable. Um, there are compounds within cruciform vegetables that need to be detoxified um, by the liver in order to to move those products on so they don't build up and become toxic. Um, that's what P450s do. So I don't want people to leave thinking, oh, these P450s are bad enzymes that only deal with like environmental toxins. No, 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 they're not. They're enzymes that are out there that are needed for metabolism of broccoli. Um, um, I think and everybody has a little different uh, sort of palate. Again, I know my P450 1A2 is very high because I do drink a lot of coffee. But
Um,
the point being that do you not have enough? Because all of these enzymes are so important. That pathway over eons of evolution has made itself so that it can adjust to almost anything. So I think when so let me just jump in here for a second. I have a PhD in biochemistry. I'm not a doctor. I don't treat patients. I do, however, see lots of patient labs and things like that. When I consider somebody to not have enough heme, that's when they're anemic and they don't make enough red blood cells. But as many, many, many people with porphyria, they're not anemic. They have plenty of red blood cells. They're making plenty of heme. So it isn't that people with porphyria necessarily don't have enough heme. It's just that the pathway isn't right, and some of those things accumulate, and that's when it causes the problem. It's very rare that you have insufficient heme for viability, I'll say.
Great. Thank you so much. Okay, the next question is, if a treatment shows the glycine uptake, would it be possible for it to help people with other types of porphyria? And not, I think this person is probably talking about clinical trial medications.
Yeah. Yep. So, yeah, yes. Yes, I think that is a good thing. Um, es- Especially if the problem is coming from the red cell, because that glycine inhibitor is a glycine uptake inhibitor from I think it specifically is known to work on red cells.
Great. Thank you. Is skin bleaching among the African descent imposes a danger due to absorbance of certain light wavelength? So I think it's asking if skin bleaching can make protoporphyria worse.
Um, I think it can, I think, yes, I think it could make you more sensitive to light when you go out in the sun, but I don't know that the bleaching process itself will alter the process of synthesis of heme that's going on within cells,
Mm,
if
okay.
that's the question.
The next question is, why are symptoms so different for different cutaneous porphyrias if they have similar reactions to the light?
Hmm. I don't know. I'll tell you, I wish I did know. I mean, for a long time, I wrestle with so let me just go back here. So I did a lot of work on PCT, right? And in PCT, you accumulate uroporphyrin. That's three. That's what you accumulate. You might accumulate some one, and some of the intermediates between euro and copro. You might accumulate some of those. Um, but it's not necessarily the levels that accumulate aren't necessarily that much higher than proto in somebody that has EPP, for example. So I wrestle with why do these people with PCT, why did they get skin blisters? Um, and why does it look like that when people with EPP, you just you get the erythema, the swelling, um, the pain, and I suspect this is not proven. I suspect that it has to do with the fact where is where is the the compound? And remember, we talked about protoporphyrin being very non-water soluble. We have another term for that. We call that um, hydrophobic. It's very hydrophobic. It sticks to lipids. It sticks to membranes of cells. Whereas uroporphyrin 3, that it's in a different place. I did a lot of work on this. So how many minutes? So we got 5 minutes left. I'm going to try to make this story short. So in PCT, it was sort of people didn't really understand exactly what that was um, until around the time that they decided to build the Panama Canal. And they were treating people for malaria prophylaxis, right? And they were giving them chloroquine for malaria prophylaxis. Malaria inhibits, the chloroquine inhibits the malaria parasite. We don't have to go into that. It does. If anybody's ever gone to Africa, you take chloroquine or hydroxychloroquine as a prophylactic. Um, and there were some people that took chloroquine and they had this, like, the next day, their urine turned bright red. They and they couldn't figure it out. Well, it was because they had PCT and all of that porphyrin that was stored in uh, in cells in the liver. When it saw chloroquine, it came out. Um, so that was a sign that it was different. And so we used to use mouse models to look at where these different compounds accumulate. And uroporphyrin actually accumulates in the lysosome. It's a vesicular body within a cell that does various things, but uroporphyrin accumulates in lysosomes, whereas protoporphyrin sticks to membranes all over the cell. And it's a long answer to a short question. I think it has to do with where those compounds are located, but that's not known for sure.
Thank you so much. So we only have 3 minutes left. So I'd like to, before we part ways, just make sure everyone's aware of other resources and events coming up for the Porphyria community. Thank you so much, Dr. Phillips, for your time. This is really, really helpful for me at least, and I hope for everybody else. So I'm going to share my screen just to plug a couple of things that are coming up with the UPA. Um, so we have our patient and family conference coming up in October. We really highly recommend anybody who wants to connect with the Porphyria community, anyone who's a patient who maybe hasn't met other patients or just wants to, be a part of the community, that this is going to be a really fun event. And there are some stipends available to help people who travel. And so we're hoping that that's going to be a really good time. And then there are also a ton of ways to get connected with the UPA. We have Connect Up meetings, small group meetups. We have one-on-one connections and a patient navigator. So if you go to porphyria.org, you can sign up for the e-news. there are plenty of links there for you to get connected and to sign up for any of our events. So yeah, feel free to do that. And I'm going to stop sharing. So yeah, so before you go, encourage everybody to subscribe to the e-news, to join us at porphyria.org. And at the end of this, you'll be asked to fill out a quick evaluation when you leave the webinar. So please, if you have the time, take a few minutes to share your feedback. It helps us develop and deliver resources that meet your needs. So thank you everyone so much. Be safe. Have a good weekend.
Thank you.
Yeah, thank you all.